So you've seen my credentials up here on the slide. One of those credentials is that I am the academic director of the ALS clinic here at UBC. It's actually housed at G.F. Strong Hospital. When I was a research fellow at the University of Chicago, in quick succession, I was seeing 3 young women who I diagnosed with ALS. All 3 of them died within a year of onset. And when I say young women, 2 of them were teenagers and 1 was a 20-year-old. So this is the sort of event that is shockingly impressive to me and everyone else. You know, no matter how much I knew, no matter how smart I was, no matter how compassionate I was, I didn't slow down their deaths by a day. All I could do was offer support. And in that tragedy was born a resolve to make a contribution to ALS and hopefully a therapeutic contribution. So 30 years later, 300 publications, 3 biotech companies, I feel like I'm starting to get close. My lab is starting to get close. So, but many of you may not even know what ALS is, Lou Gehrig's disease, as John was saying. So I also work in other neurodegenerative diseases, and I just wanted to put up a slide about Alzheimer's, not so much to explain the pathophysiology, but to, you know, kind of like sketch out what this disease is doing to individuals, families, and nations. You know, this is also a shocking statistic that every 4 seconds somebody in the world is diagnosed with Alzheimer's disease. There is no effective treatment or a disease-modifying treatment to slow it down. There are symptomatic medications that can make somebody more comfortable and functional, but it's still a tragedy to be robbed of your cognition, your personality, and everything in this inexorable progression. So another statistic I like to cite is that in Canada and in the US, and in fact every country in the world, at least every developed country, the onslaught of dementia, particularly Alzheimer's, is going to break the bank in our health systems. At present, we spend about $20 billion a year managing Alzheimer's in Canada. The US appropriately spends 10 times as much with 10 times the population. In just a few years, this will surpass the budget of healthcare in both countries. But it is amazing to realize this last statistic, the one over in the corner, which is if we could even delay the onset, not prevent it altogether, but delay the onset by 5 years, now we would save about $5 billion in Canada. So this is a very serious problem for individuals, their families, their communities, and their countries. So now I'm going to talk to you about these molecules that I'm so fascinated by, having spent a research career studying them. But I want to show you something that you may not know. So I'm going to try to linger on this slide and just to kind of teach the amazing changes that are happening in my field of neurodegeneration. So on the left is a protein, and it doesn't look like a protein, but it's a chain or a string of amino acids. Those amino acids are encoded by the DNA and by the RNA, and finally they're translated in the cell to make an immature protein. Now that floppy string on the left is the protein, or at least it's the sequence of the protein, but it can't perform its function without being properly folded, and that's the middle panel, a nice folded, patterned, three-dimensional structure which is necessary for a protein to do its job, whether it's cytoskeleton to support the cell, to transfer molecules from place to place in the cell, to function as an enzyme, to function as a receptor. All these proteins need to be properly folded in order to do what they were made to do. And that used to be it. When a protein wore out or became oxidized or fragmented, it was recycled into its constituent amino acids and just goes over and over again. But in the 1980s, there was a scientist, there is still a scientist named Stanley Prusiner, who developed a hypothesis to explain a weird type of infection. And these were called the transmissible spongiform encephalopathies. The human disease is called Creutzfeldt-Jakob disease, but mad cow is also a transmissible spongiform encephalopathy. What he guessed, the inspired guess that is a hypothesis, is that the infectious particle for prions did not contain a nucleic acid, didn't contain DNA or RNA. What it was a kind of misfolded protein that could recruit more copies of the normal protein into itself. So it was acting as a template to convert normal protein until it basically gobbled up all the protein in the cell, all the protein of that particular type. And there was a loss of function of that protein. The misfolded protein also had toxicity, and it was the perfect storm for the brain in which this process actually induced a transmissible spongiform encephalopathy. So Dr. Prusiner won the Nobel Prize in 1997, basically only about a decade after he first made this hypothesis. But the rest of us dealing in neurodegeneration, we're wondering, golly, I wonder if this prion principle could also apply to the diseases the common diseases we know and hate, like Alzheimer's, like Parkinson's, like ALS. So labs around the world, including my lab, began implicating proteins in this process, in the propagated protein misfolding that underlies the prion diseases. So I'm giving you a very simple example of how we might handle, this challenge of a prion-like propagation of a protein. There are two nice healthy cells. In one of the cells, a protein converts into a misfolded form. Because it can template other normal proteins, the other proteins become converted. That can get expelled from the cell, transmit to a neighboring cell where it's taken up, and lo and behold, the same process is initiated in which all of this particular protein in all of the cells becomes converted to a misfolded form, and both cells die. But what if we were able to block these prions in between cells while they're transiting from one cell to another? And here's the same beginning process. that one cell, all the protein is converted. The misfolded protein gets expelled and is about to be taken up by a neighboring cell, and we run in there with an antibody which blocks the infection, so to speak, of the neighboring cell. So this is a big idea, one might say, but it has its detriments, including the fact that if you just raise an antibody against a misfolded protein, it's going to react with not only the misfolded protein, but also the normal protein. So we in the lab have spent about 15 years trying to figure out a way to drug this situation, to attack a misfolded protein while sparing the normal protein. And we've been successful for several proteins. Exemplars are here. The original prion protein itself, this is the one associated with transmissible spongiform encephalopathy. There's a vaccine in development by a group in Saskatchewan that are using it to prevent sheep scrapie and chronic wasting disease, which are two prion diseases in agricultural animals and in the wild. The middle on the right side is one of my favorite molecules, Superoxide Dismutase 1, known by its friends as SOD1. And this protein, we were able to map parts of the protein that become exposed when the protein is misfolded in disease. So this is a way of specifically attacking the prion-like form of SOD1 while sparing the normal protein, which protects us against free radicals from damage or neutralization. On the bottom is our first effort into developing antibodies which are specifically directed at toxic oligomers, and I won't even go into that, but oligomers are small clumps of proteins, and this particular oligomer is implicated in Alzheimer's disease. So we now are in a position where so much has come out of the prion hypothesis, not only for ALS, which is what I started with, but for Alzheimer's disease. One of our anti-oligomer antibodies is going into clinical trials for Alzheimer's disease at the end of next year. There is technology for ALS that's waiting to be licensed. And we're also approaching a specific misfolded specific antibody for Parkinson's disease. But the universe is expanding, and Huntington's disease has a prion-like component. Type 2 diabetes has a prion-like component. This was published last year to the astonishment of many people in the field. And even schizophrenia could be using the prion process to make nerve cells dysfunction in the way that produces psychosis. So I'm going to stop there, but I want to point to my wonderful collaborators, not only at UBC, but in other parts of Canada and in fact around the world. I want to express my gratitude to the funding agencies which have supported some of this work, and particularly I want to express my gratitude to the philanthropic donors who have provided funding for critical steps in this process.